Thank you very much. CTAF is back in its seats. Mr. Chair, with your permission, I think we're ready to start for the voting and deliberation phase. I just want to let Laura please come up and make sure that the CTAF panel is hooked into the voting system and we have a chance to do a test of that.
Polling closed.
All right, welcome back, everyone. We're going to run through a quick test vote to make sure that everyone's software is running properly. If you don't see this screen on your laptop or phone, just raise your hand and Monica will come by. So for this question, we're looking for 14 responses. Though today the Marriott is known as a hotel, in 1927 the Marriott was known as what? A root beer stand, a travel agency, the apartment that housed the Oakland Athletics baseball team, or a pharmaceutical company. So I'll give everyone a few minutes to vote. If you're having any issues, again, Monica can come by. 4 more responses. Do you have another remote? Okay, we're looking for one more. I think it's Rita. We're all friends here. No.
Tension is killing us for you.
I know. I've been studying this all week.
The stakes are very high. All right. The correct answer is A, a root beer stand. All right. Well, thank you, everyone. I'll pass it back over to Steve.
God. I say this every time, but it's true every time. That is the most interesting part of this meeting, right there. Right there. All right, we'll think about that. All right, we have until 2 o'clock by the agenda. If we finish early, we may shift earlier into the break and the policy roundtable to give us a little bit more time. But we have an hour and 15 minutes, and I know that you've already covered some of the issues around the clinical evidence, et cetera. But this is your chance again with the full input from the clinical experts and patient experts and with the ICER and UCSF team up there to think through again some of the issues around the votes. So the votes are structured first with an emphasis on comparative clinical effectiveness votes. And for the first 6 questions, the patient population is the following. Adults ages 18 or older with moderately to severely active rheumatoid arthritis on conventional DMARD therapy who are naive to TIMS. With that in mind, the first question is, in patients who are naive to TIMS, is the evidence adequate to demonstrate that the net health benefit of upadacitinib plus a conventional DMARD is superior to that provided by a conventional DMARD alone? Jeff, could you just briefly summarize again the evidence and the ICER evidence rating and refer to the tables in the evidence summary for CTAF?
Sure, Steve. So for those who want to see the evidence ratings, they're all on table 1. But for this question in particular, tables 2 and 3 have the randomized trials of upadacitinib in this patient population. Table 6 is the network meta-analysis results, and then table 13 are the harms. And table 13 has the harms for all of the different comparisons. So you'll always be referring back to table 13, though as you'll see, there aren't big differences in the harms across the different medications. So the bottom line here, we gave it an A because when you compare to placebo, you see much greater rates of achieving low disease activity by whatever measure. So if we use SDI, it's 40 versus 15%. If we use CDI, it's 40 versus 16%. It's not dependent on the DAS28 CRP, you see markedly greater percentages of patients achieving at least low disease activity with these. And when you look at remission for the DAS28 remission, for instance, 29% versus 6%. And that's true also for the ACR outcomes, both in Table 3 and the network meta-analysis results. So multiple trials, large, substantial clinical benefits, harms that are real but relatively small. So high certainty of substantial net health benefit was our assessment.
Okay. And again, just to demonstrate, to reemphasize that this is among patients who have been on conventional DMARD therapy and who still have moderately to severely active RA, despite that conventional DMARD. Any questions about the thinking? Say yes?
On Table 13, Jeff,
sorry, can you just talk a little bit more?
Sorry. On Table 13, Jeff, could you speak to the rates of any infection and kind of the profound differences between some of the comparisons there, or the differences in definitions?
I actually pointed that out to the staff. I worry that there's a typo there. I wanted to go back and look at that. So I think what Dr. Lee is talking about is the any infection rate for, is relatively low for the three JAK inhibitors. It's 0.5, 0.4, and 1.4, and yet for adalimumab it's 41.9%, which looks like a typo to me, and that's what I'm concerned about. Even the CDMARD plus placebo in the trials, they were fairly similar. So I think there's a typo in this table. I'm sorry. I think that's an error.
Let's pause to flesh that out. Serena, can you go back to the table on harms and?
So it's looking at the any infection and the serious infection rates.
That would be very surprising if those numbers were real.
Yeah, I highlighted that this morning as well. In general, the serious infection rates look correct. I mean, they're similar across the different classes. It may be that for adalimumab and the CDMARD plus placebo, where the N's are so much bigger, it may be some older trials were included, and not the trials with JAK inhibitors. But it still looks so there may have been older, longer trials that contributed to that. That may be the problem, length of follow-up in those.
Just on the line right above that, Jeff, what are your thoughts about the discontinuation due to adverse events? I'm kind of surprised by how high. It seems like the adverse events, serious adverse events, seem pretty comparable between, but the discontinuation due to adverse events is
a little bit higher in Tofacitinib.
Hard to know.
Yep, I agree.
Of course, we don't have the statistics to show what the precision is around those estimates. My guess is with a very low rate. Jeff, do you have any memory?
Yeah, no, they're not significantly different, and certainly in the head-to-head trials, they weren't significantly different. Steve.
Other questions? Kim? Well, we're continuing to look, by the way, for that back in the weeds of the report. Yes, go ahead. Sorry, can you pull the mic closer, please?
On that same table, any adverse events, like it's over half the patients?
That's pretty typical in most clinical trials because it's like colds and flus. They're not necessarily related to the therapies. Okay. So often in other longer clinical trials, you often see 80, 90%, and it's not related to the drug.
Thank you.
Rita.
That actually reminded me, I know we're looking at 1 year, but because there are these immune modulating effects, there's always concerns about cancers long-term. What data do we have on cancer risk?
Very little. I mean, as we heard earlier from the clinical experts, there doesn't seem to be a dramatic increase at years 4, 5, 6 in the few patients who are on either an individual therapy for the long-term or patients who cycle through multiple of these immunomodulatory therapies. But I think there's a signal that there's an increase in lymphoma, hence the black box warning, but the absolute incidence remains low.
The other comment on that would be that it's become clear that rheumatoid arthritis itself is, for many of these patients, the primary risk factor for hematologic malignancy in particular. And so as they try to control for disease activity, it seems the more active the disease over time, the more likely that is to occur. So there's controversy in the field even as to whether there is a link at all. And I'm speaking to the TNF antagonists primarily since they were the first drugs to have that warning. But having said that, with those drugs, there's no signal over time of increased risk.
And how is that teased out? Because I imagine the more active the disease, the more treatment you get.
Right. I believe it was about 15 years ago, there was a study in the New England Journal that made that link, and that was kind of the black box stuck at that point. And then most of that is being done through registries, primarily in Europe, looking retrospectively at large cohorts and trying to control for disease state, biologic use versus not. And no one, to my knowledge, has reproduced that signal in the last decade with the TNF antagonists. Again, that's different from the JAK inhibitors.
Okay, I think we are going to go on the assumption because we're still looking backwards, but I'm going to suggest that we move forward with a vote with the assumption that those are typos or that there is some other rationale that would suggest that any infection, certainly serious infection you see, is the same. But for some reason that any infection line for ADA and for CDMAR plus placebo just do not seem right. So I would discount that in your thinking. We will continue to look for it, but I think we should probably move forward under that assumption. You got from the clinical expert world, that doesn't ring true, right? You wouldn't think that any infection rate with CDMAR plus placebo is 5 times what it is with any of the JAK inhibitors or the TNFs?
No, not at all. It is fairly clear that varicella zoster is increased in those treated with JAK inhibitors. That's about the only meaningful difference from an infectious perspective that I'm aware of.
Okay. All right. Why don't we go ahead and vote on this one then? We have 14 votes.
Have something? They were just raising their hand.
Go ahead.
It's a typo.
It's a typo.
Where it's a composite, so I don't have the actual number.
So again, I think pretty clear across the board. The evidence is there's not evidence that there's a significant difference in infections across all of these classes, either placebo plus CDMARD, ADA plus CDMARD, or the JAK inhibitors.
There it was. Okay, so you saw in a flash of light there, and I'm not going to go back because it'll mess it up. It was 14 yes and 0 no. Sorry, for some reason there was a time delay in showing it up here. All right, so that the answer was 14 yes and 0 no to question 1. All right, question 2 is really the same question about, I'm sorry, it's not. This is one is comparing upadacitinib to adalimumab. So let me read the question again. In patients who are naive to TIMS, is the evidence adequate to demonstrate that the net health benefit of upadacitinib plus a conventional DMARD is superior to that provided by adalimumab plus a conventional DMARD? Jeff?
Yeah. So again, the same tables, 2, 3, 6, and 16, but now you're comparing UPA to adalimumab versus UPA to the essentially placebo or conventional DMARD therapy alone. So here the differences are smaller. So again, using CDI or SDI, it's 40% with upadacitinib, 30% with adalimumab. Those differences were statistically significant. They're a little bit bigger if you use the DAS28 CRP, which again may exaggerate the effect because of the specific activity of the JAK inhibitors on the IL-6 CRP pathway. But you see clinically and statistically significant effects, but that are smaller than those observed between upadacitinib and placebo. Similarly, for the ACR 2050 outcomes, the HAC-DI, I will say
Jeff,
we don't
Jeff,
have...
sorry, you're being admirable in your brevity and how fast you're going, but I think it'll be helpful if you slow down a little bit and tell them which table you're looking at, which column, so that they can follow along with the numbers that you cite.
Great. So the initial comments were table 2, the last 2 columns where we have CDI and SDI, low disease activity, the percentage of patients. So in both upadacitinib plus methotrexate, You had 40% of patients achieving at least low disease activity by both of those measures. Adalimumab plus methotrexate, it was 30%, and those differences are statistically significant. If you look back 2 other columns, the low disease activity by the DAS28 CRP was 45% versus 29%. So again, a little bit bigger. This may in part be explained by the activity of the JAK inhibitors on IL-6, which influences the CRP level. So you may want to think about the CDI and SDI responses rather than the DAS28 CRP. And then similarly, table 3, you can look at, say, the ACR 70, so the percentage of patients who had a really good response, 25% versus 13%. Those who had an improvement in their functional status by the HAQ-DI, the change was 0.6 versus 0.49. So again, smaller than the differences between upadacitinib and placebo, but still, these are just to give some perspective on the HAQ-DI, it's a score that goes from 0 to 3. So these are clinically important differences. Those are probably the main clinical benefits. I was going to say it's not here in the tables, but again, in the Select Compare, which is the main trial comparing upadacitinib to adalimumab, there were significant improvements in fatigue, in pain, in stiffness for upadacitinib compared to adalimumab. Smaller, again, than versus placebo, but statistically significant on those outcomes that matter to patients. The harms are in Table 13.
Okay.
Again.
And the preliminary evidence rating given to this was a B+, correct?
It was a B+, yes. So smaller trial, the differences are smaller than in than versus placebo, so a little more uncertainty, and so we said small to substantial net health benefit.
Okay. In some discussions with clinical experts, there's been some questioning around whether the patient selection for the trials for UPA and adalimumab were somehow tilted, not consciously perhaps, but tilted in some way that might highlight the potential clinical superiority of upadacitinib. Would you reflect on that again for the panel?
Sure. So do you believe in the grassy knoll, and do you believe in Sasquatch? So there was a trial called the Balance 1 trial, which was a phase 2b trial of upadacitinib that used as enrollment criteria either elevated high-sensitivity C-reactive protein or seropositivity for both rheumatoid factor and anti-CCP antibody. In analyzing the Balance 1 trial in a separate paper that was a very strange paper that was really the mechanism whereby AbbVie converted a BID dosing regimen to QD for the phase 3 trial, there's specific mention made of the fact that, I'll read it, of the covariates evaluated, only baseline high-sensitivity C-reactive protein showed a statistically significant correlation with the upadacitinib exposure-response relationship. Higher upadacitinib maximal effectiveness was estimated with higher baseline high-sensitivity C-reactive protein among a group of patients who had failed TNFs. The concern then is that when you look at the Select Compare trial, all of those patients they were required to have an elevated high-sensitivity C-reactive protein for enrollment. So at first blush, this might look like the well has been tainted, that we've only selected a group of patients with high-sensitivity C-reactive protein to bias the results in favor of upadacitinib over adalimumab. It turns out that it's quite common to require an elevated C-reactive protein for enrollment in clinical trials. The problem really gets at the fact that C-reactive protein serves as both a diagnostic marker, as a marker of severity, as a marker of responsiveness, and in this case, as a marker for responsiveness. It's unfortunate that that same marker has all those functions. It should be noted, though, that in populations that were methotrexate inadequate responders, the high sensitivity C-reactive protein did not fall out as a predictor of response to upadacitinib. It's only in TNF non-responders. And the Select Compare trial is made up of those that are methotrexate inadequate responders, in essence, as you follow through the study. So there's some reason not to be as concerned at first blush, but I will say this. In Select Compare, they went on to flip treatments. If you didn't have an adequate response to upadacitinib, you flipped to adalimumab. If you didn't have an adequate response to adalimumab, you flipped to upadacitinib. That analysis, to me, is completely tainted by the fact that going into the study, we only included a group that if they don't respond to adalimumab, they're more likely with a high C-reactive protein to respond well to upadacitinib. So it's a nuanced, complicated, convoluted series of considerations here. But in the end, I think with some caveats, we can say that at least in the population who has an elevated C-reactive protein at baseline, which is not all the patients that we see in the real world, at least in that population, it would appear that upadacitinib is performing slightly better than adalimumab. I have some real concerns about whether this can be generalized, though, to all the patients with rheumatoid arthritis that we see in clinic. And there are tremendous differences between clinical trial populations in this disease and real-world patients that show up, to the point where if you look at registry data from the real world, some 80% of patients in real-world rheumatoid arthritis registries would not qualify to enroll in the clinical trials that have been done to approve the drugs that they're being treated with. So I think all of this should provide perhaps some pause in the certainty with which we approach the Select Compare trial.
I hope you followed that. You did? I know you did. Jeff,
yes.
Reflect.
Yeah, the only other thing I want to say related to that is the two trials of tofacitinib that included adalimumab arms, which are oral standard and oral strategy, they had slightly different entry criteria. One had CRP greater than 3, one greater than 7, and they also had sedimentation rate entry criteria. But the mean CRP level in those two trials and in Select Compare was 17 or 18. So they ended up with populations with fairly similar degrees of inflammation as measured by CRP anyway. So it shouldn't color, I don't think, or at least comparisons across those trials.
Okay, so at the end of the day now, CTAF is going to take this vote on UPA versus, sorry, I drop into shorthand, upadacitinib versus adalimumab. B plus was the preliminary rating. Do you see any reason to shade that for the CTAF panel from your perspective?
Not based on the CRP entry criteria, no.
Okay. Questions from CTAF? Who's going to vote, anybody going to vote no on this, want to share their thinking with panel colleagues? Saleh, you want to give your rationale and let people hear it?
Yeah, I mean, I was kind of struck by Dr. Konkoff's statement about how if the statement was for the population where the data is coming from, then I think it would make sense. So instead of saying in patients who are naive to TIMs, if we're talking about if this is for patients who have this specific HSCRP, meet the CRP criteria, then I would feel much more comfortable. I'm also, the second line thing that I'm really grappling with is I'm really bothered by how many more people in the UPA arm dropped out due to adverse events. And we don't know much about what those adverse events are, but I feel like people are voting with their feet not to continue to take this drug. And so I really, that is, it's maybe it was not significant, but there's at least a trend that's concerning. And that, along with this question, is enough for me to say I don't have the level of certainty to say that this is clearly superior.
Any other questions or thoughts about that? Jeff, any reflection on that from your perspective?
No, I wish I had my documents in front of me just to check, particularly in Select Compare, what the dropout rates were to see if there was a significant difference between adalimumab and upadacitinib in that trial, which is the only head-to-head. Because the adverse event rates are sort of pooled across all the trials. But I don't recall it being that big, but I don't have that right in front of me. So,
Serena, can we grab that, do you think, or not? So give us 2 secs on that, or maybe Laura already knows.
It looks like it was a difference of like 3.5 for UPA versus 6.1 for adalimumab.
Yeah. So in the trial, that was just handed to me. AEs leading to discontinuation was highest on adalimumab, and the overall AE rates was higher on adalimumab.
So that's interesting. In the head-to-head, then you're right, it was flipped.
And so the 8.3%.
Sorry, can you grab your mic just to make sure everybody on the webcast can hear you?
So the 8.3% on UPA and 2.9 on adalimumab are from different studies and different
it's sort
populations.
of the pooled UPA data, across all the different trials, so they're not necessarily head-to-head.
Okay. All right, go ahead and vote. Just waiting on one more. There we go. All right, see if I can do this one correctly this time. I don't know why it's doing this. Hopefully it won't skip through again. There we go. I'm not going to touch it again. All right, 12 yes and 2 no. All right, because I think we had a good chance to talk about the issues before the vote, I'm going to move forward. The next question is, again, same patient population, patients who are naive to TIMS. Is the evidence adequate to demonstrate that the net health benefit of tofacitinib plus a conventional DMARD is superior to that provided by a conventional DMARD alone? Jeff.
So here we're moving to tables 4 and 5, which are specific to tofacitinib in this group. Table 6, again, is the network meta-analysis, and then table 13 are the harms. And here for tables 4 and 5, you want to focus on the comparisons between tofacitinib and placebo for this question. And you see for most of the trials, we have not reported. That's because we emphasize the 3-month results in our analysis. However, there are, as I said, there's a publication that pooled all of the tofacitinib trials versus CDMARD alone in this population, specifically in the TIM-naive. They broke it out in that publication between TIM-naive and TIM-experienced patients. And you see big differences. SDI, the last column, 35% versus 14%. CDI, the second to last column, 32% versus 14%, and so on. That's in Table 4. Table 5, the 3-month data for the ACR outcome were reported consistently at 3 months. For instance, in the first trial, oral standard, the ACR 70 column, it's 12.1% met that criteria versus in the tofacitinib arm versus 1.2% in the placebo plus CDMARD arm. And you saw similar results across the board. In the pooled data, it was 12.9% versus 2.8%. So large differences compared to continuing on CDMARD therapy alone. Harms, relatively modest, some important significant harms, but relatively modest. And so we gave it an A rating, high certainty of substantial net health benefits compared to placebo again.
Okay, we've been through some of the issues already, but this is obviously head-to-head versus placebo with continued CDMARD use. Sorry, bless you. Any other questions? Okay, please go ahead and vote. Oh, very good. All right. Come on, this one's going to work. Oh. I believe it was 14-0. Anybody else see the same? Yes. Okay. This is a test of your eyes. Clearly, if it's a test, I'm failing it, but we'll try to get that. All right, so we're going to move on to the next question. So just again, for the record, 14 yes, 0 no for tofacitinib versus continued CDMARDs. All right, the next question is, in patients who are naive to TIMS, is the evidence adequate to demonstrate that the net health benefit of tofacitinib plus a conventional DMARD is superior to that provided by adalimumab plus a conventional DMARD? Okay. Jeff, here we don't have head-to-head data, so lead them through what we've got.
We actually do have some head-to-head data.
Thought we did.
Again, we don't have the 3-month data for the outcomes here. Actually, we don't have 3-month data except for ACR. So table 4, we don't have anything. Table 5, we do. So for in the oral standard trial, the first trial, those who achieved ACR 70, it was 12.1%, as we said before, versus 8.4% for adalimumab. In the oral strategy trial, that was 19.3% versus 14.3%. And also, we have the network meta-analysis results. And if you remember in the figure, which made it easier to see, the bars for tofacitinib and adalimumab were essentially the same height for those who were less than 20, 20 to 50, 50 to 70, and 70 to 100. And in the conclusions for both of these trials in the publications was that tofacitinib was either non-inferior or equivalent to adalimumab, and there were no important statistically significant differences on any of the measures in either of the trials. So my conclusion was, I gave that a C, that tofacitinib is comparable to adalimumab in this population. I should say the trials were mainly used, as we heard, because they were older trials, they were based in when the mindset was more a 6-month outcome time frame. So the primary outcomes were 6-month outcomes. And again, at 6 months, there were no significant differences between the two therapies.
Okay. Patient experts or clinical experts, any perspective that you want to bring to the data?
I would agree. Looking at these studies, the differences appear to be fairly small between the two arms.
Okay. Questions? Yes, Annette.
For the clinical experts, I have a question. So is it biologically plausible that one JAK inhibitor would be better than adalimumab, but the other one isn't? Or do you think this is a difference in how they conducted the study and how they enrolled, what the patient selection criteria?
Yes. I think the manufacturers learn how to design studies that get the outcomes that they're interested in, obviously, right? There is enough biological difference in the specific targeting of these drugs that it's certainly plausible to see differential outcomes, I would say.
So this gets back to whether you believe selectivity can confer a efficacy advantage or not. Tofacitinib is selective for JAK1. Tofa is 1-3 at least, plus possibly others. If you believe that that can confer a benefit, absolutely there can be a difference. But I think it's important to understand that these studies have been conducted in different eras from a understanding of how to massage the data to datasmith toward what you're looking for. So I wouldn't sell short our pharma colleagues' ability to maneuver populations. So clearly, Tofacitinib research scientists have had all the years that intervene to do their best to put their drug in a more favorable light.
To that end, so are you saying that, how hard was it to recruit the, I'm going to get the names all mixed up, the U1, upadacitinib trial in the light of the fact that there are already JAK kinase inhibitors on the market and TNF-alpha drugs are widespread? Are they going to countries outside of the Western Europe and the United States to recruit these patients, or how are they able to do this?
I'm not sure where these particular studies were conducted. I think these were primarily domestic studies. I know some of the upadacitinib trials were.
I think, so like compare, I think 27% were in Eastern Europe, and then the next largest was in Central and South America. So the minority actually was Europe and North America, I believe.
Sure.
I may be wrong on that, but I'm pretty sure that's correct.
Speaking to conducting RA trials in general, it's become increasingly ethically murky to have the placebo arms given the effective drugs that we have. And that is, for me anyway, having done some clinical trials, that's been kind of a bugaboo, so to speak. So, they get around this by trying to have short placebo arms with crossover to, to some treatment if you're an inadequate responder. Probably a lot of these trials are enriched for patients who may be economically disadvantaged and may not have access to some of those other treatments as well. Yeah.
I just want to make a broad kind of framing suggestion around some of the comments here, and I do not want to keep us from talking about evidence and even assumptions. But I think given that we don't have a setting in which the drug companies can respond immediately to some of the things that are being said, it's fair just to I think, assume that we don't want to assign intent necessarily. We can raise questions for people to consider, which I think is very fair. I want to frame our comments in that way, that we're raising considerations about the context in which these trials are done, both historical, evidence-based, and incentives are on the table, but just want to frame it as a question raising for the CTAF panel to consider. Yes?
So then my other question is, because to me it would seem that non-selective might have more efficacy, maybe also more toxicity than a purely selective, but maybe I'm wrong on that. And unless you think it's an issue with dosing, so were these drugs adequately dose ranged, and do we know that these are the T drug, this one, Tofacitinib, is if that's the difference between this one and the other one?
So I don't necessarily think that's the case, because if you're particularly selective for JAK1 and JAK1 is mediating the effect, you may have a better inhibition of JAK1 relative to the others. And so I think there's a lot of complexity that may go into that calculation.
Any other comments or questions? Okay, thank you for that input, and let's go ahead and vote. So this is Tofa, and again, I will use the shorthand because even though I try to say it longhand, sometimes I trip too. Tofa versus adalimumab. All right, finally a test for me. There we go. It worked. Okay, 14 no and 0 yes. All right, so onward we go. Question 5. In patients, we're back still with patients who are naive to TIMS. Is the evidence adequate to distinguish the net health benefit between upadacitinib and tofacitinib? Jeff, how would you frame the evidence on that?
So here we have no head-to-head trials, and we were challenged to do indirect comparisons. The only data really come from Table 6, which is the network meta-analysis results with the ACR criteria, where you see a slight advantage to upadacitinib compared to tofacitinib, not statistically significant. And then you have the adverse event data in Table 13 again, but different populations, different lengths of follow-up, so hard to compare when you don't have head-to-head. So we gave this an I rating, insufficient evidence to really judge if there's if there's a difference in net health benefit between the two.
Clinical experts, others, anything to add? Do you want to add? Don't need to. Okay. Questions? All right, please go ahead and vote. All right. 14 no, 0 yes. All right. So with that, then we will skip question 5A, which was going to say which of the two was better. Since we cannot distinguish between them, we will go to question 6. Ta-da. Okay. So, shifting orbits here, we are now going to talk about biosimilar. And I will also emphasize what Jeff said, which is we face the challenge of not being able to do a report on every single biosimilar in the autoimmune space. We decided to pick one, not because it's the best, the worst, an average. It's meant to be an exemplar for us, biosimilar exemplar. Got it? Got it. So it's meant to help us examine the issues around biosimilars, both their evidence and also the policy aspects. So with that as framing, we are going to ask you about this one. Is the evidence adequate to demonstrate that the biosimilar infliximab, DYYB, produces a net health benefit comparable to that of the originator biologic infliximab? Jeff?
Great. So here we do have a head-to-head trial. It's required by the FDA. The summary of the results of the trial are in Table 14. The primary outcome was the ACR 20 measure, which has typically been the primary outcome in many of the FDA-sponsored trials. So the ACR 20 outcome at 30 weeks, and you see that they were very similar, 60.9% for the biosimilar, 58.6% for the reference drug. So if anything, slightly favoring ACR 20, not statistically significant. If you don't like ACR 20, they got ACR 50, ACR 70. They got DAS 28 with CRP or ESR, SDI, CDI. All of those are recorded at 30 weeks, and there are no significant differences, and you see numerically they're very, very similar. Adverse event rates, again, here we have head-to-head, so you can see they're very, very similar. A few more serious adverse events in the infliximab DYYB, but that's, again, these are not necessarily adverse events that are related to the therapy. They're just all hospitalizations and other serious adverse events. The infection rates, God, no, this is one, the any infection rate clearly is wrong, so I think this is a typo because we have a higher serious infection rates than any infection rate, so disregard that. They were really comparable across the drugs. And then, as I said in the evidence presentation, there are data at 54 weeks and 102 weeks, and the results were similar for these measures as well as measures of joint erosions using the joint erosion scales.
Good. Any questions about the data here, the evidence? Anything to add?
Yeah, I would simply add this is the tip of the iceberg as far as these studies are concerned. There are several other, particularly European studies, showing similar outcomes with this and other infliximab biosimilars.
All right. Okay, thank you. I'm not seeing any cards raised, so please go ahead and vote. Is the evidence adequate to demonstrate comparable net health benefit for the two. 13 yes and 1 no. All right. Since we had a no, just might as well real quickly, anybody want to say why, or was it a possibly, it's always possible it was a mistyped vote? Did somebody mean to say that they didn't think the evidence was adequate to support comparable net health benefit? No, I'm wondering if it was a typo, if you know what I mean. We've had a few of those so far. Can you see your votes?
It tells you what you
It tells you what you voted. So somebody voted no. Can you check your vote? Okay, well, we'll assume that somebody intended to vote no, but chooses to remain anonymous. That happens. That happens. All right, let's keep going. Okay, we are now shifting to Tim Experienced. And I want to try, we're doing okay actually with the pace here. So for the next 3 questions we have now going again to adult patients, still with moderately to severely active RA, who are on CD-MARDs and are now Tim Experienced and continuing to have moderately to severely active RA. For this patient population, is the evidence adequate to demonstrate that the net health benefit of upadacitinib plus their ongoing conventional DMARD is superior to that provided by a conventional DMARD alone? Jeff.
So here, and this will be true for the next 2 questions as well, we have just one relevant trial. So for upadacitinib, it's the Select Beyond trial, and it's summarized in tables 7 and 8. And again, we didn't break out, maybe in retrospect we should have, didn't break out the harms by Tim Experienced or Tim Naive, but it's the same table 13 of harms. But again, in general, there really was no signal of differences in adverse events with these therapies. So for, I'm starting in the column to the right in table 7, you see a substantially greater response with upadacitinib than placebo, 34% versus 14% using SDI, 32% versus 14% using CDI, and so on. Slightly greater difference with the DAS28 CRP. And the absolute difference is smaller than that seen with the Tim naive patients, and there's plenty of clinical experience backing that up. Once people have failed therapy, they don't get as big a response to an additional therapy, but you do have some response. Because it's one trial, the effect size is a little bit smaller. The sample size is, at least the net sample size, is smaller than in the Tim naive patients. I felt there was greater uncertainty. The benefits were smaller, so I gave it a B+. So moderate certainty of a small to substantial net health benefit compared with placebo.
Okay, any questions? You guys are getting pretty fluent with the data now. All right, please go ahead and vote. Okay, 14 yes, 0 no. Thank you. The next question, boy, that responds really quickly. Wow, I've even learned how to do that. All right, the next question is still Tim Experienced, and now we're going to asking whether the evidence is adequate to demonstrate the net health benefit of tofacitinib plus conventional DMARD is superior to that of DMARD alone.
So now we move to tables 9 and 10. They're analogous for 3-month outcomes for tofacitinib. It's one trial, the oral step trial, plus pooled trials. Again, most of the other trials, they didn't present the breakdown, but they did pool the data at 3 months for CDI, SDI, DAS28, and so on. So it may be most useful to look there at the last row in both of the tables. So 30% versus 14% for SDI, 29% versus 14% for CDI, and DAS28 ESR, in this case, low disease activity, 13% versus 5%. And then the next table, table 10, looks at the ACR results and the change in the functional status measure, the HAQ-DI, where again you see significant differences. Similarly, one trial a little smaller than in the TIM-naive, smaller absolute benefits than compared to placebo in the TIM-naive population, so a B+ rating as in the prior example.
Okay. Clinical experts, are we missing anything here from your view in the discussion? Okay. Any questions? All right. Please go ahead and vote. Oh, I keep renewing it. That's why. There we go. All right. 14 yes and 0 no. All right. Into the home stretch on the clinical effectiveness questions. We have one more. In patients who are TIM experienced, is the evidence adequate to demonstrate the net health benefit of Baricitinib plus a conventional DMARD is superior to that provided by a conventional DMARD alone? Baricitinib finally enters the arena. Jeff?
I was going to say this is the first time we have Baricitinib in a question because, again, it doesn't have an FDA indication for the TIM-naive, only in the TIM-experienced population at this point. The analogous tables are tables 11 and 12. One trial, the RA Beacon trial, and again, in table 11, 22% versus 9%, 24% versus 11% for SDI and CDI, respectively. For low disease activity using the DAS28, they had reported both CRP and ESR, 24% versus 9% or 13% versus 4%. You see the DAS28 CRP tends to give a higher response rate than the DAS28 ESR. So similar sorts of findings, similar also by the ACR metrics 20, 50, and 70, consistently better than placebo and statistically significant, relatively modest effect sizes. So we gave it a B+ again, like the other two.
Okay, and the harms, the harms track.
Yes, I'm sorry, that was the one thing I was going to say. You may want to flip to table 13. This is the first time we've really looked carefully at Baricitinib in terms of harms. But again, they look fairly similar to the other JAK inhibitors, so there's nothing that really stood out in the clinical trials.
Clinical or patient experts, anything we should be focused on in particular as we look at the data here? No? Okay. All right, go ahead and vote. Oh, I'm sorry.
Again in the table?
Sorry, can you use your mic?
Is there a typo in that table again, the 9.4, any infections?
And also the 41.9.
Yeah.
Clearly the 41.9 was infection. Was a problem. I think this, again, reflects, partly there was a typo in the adalimumab, but I think partly it reflects the fact that there are different patient populations. So this is not specific to the RA Beacon trial. This really is across all of the trials of Baricitinib. So I think it doesn't fairly reflect the comparisons here. I think clinically we know there isn't. I think in the RA Beacon trial there were no important differences.
Yeah, and again, I haven't looked at the primary data, but the serious infection line being the same, it's possible that some of the Baricitinib data, they just either were more inclusive of common colds or other kinds of infections, or had some other feature that made any infection a little bit higher. It's hard to know.
We looked back at the data, and the number that's 41.9 should be 36.1.
So it's still high.
Yeah.
For adalimumab?
Yes.
So the bigger issue is the length of follow-up for this. So if this is the absolute number, not the incidence rate. So it's a percentage, and I think the adalimumab trials were longer. So this is somewhat misleading because of that. Sorry, Steve, I see you shaking your head.
Okay. So is it possible that the other two are typos? Because they're exactly the same numbers, any infection, serious infection for TOFA and for UPA? that all infections were serious infections? That doesn't quite make sense. Because
Right.
I think zoster was brought up. Yeah.
So, I mean, just in the interest of clarity, can we bring up the RA Beacon trial and just look at that, because that's most germane to this particular question?
I will look up RA Beacon, but I will say that for upadacitinib, any infection should be 30.7, and serious infection should be 1. So that is a typo, and I'm going to take a look at RA Beacon. Thank you.
You're right. The anti-infection is higher.
Well, that clarifies everything. Jeff, try to summarize now, again, your take on how the panel should address the harms data as it's evolving at this point.
So this table is probably not the best table to, because it sort of pools adverse events across multiple trials, multiple lengths of time, different indications, different patient populations. So this is probably, it was not the best table to include for adverse events. I will say in the randomized clinical trials, when you look across them, the adverse event rates were very, very similar in the arms of all the trials. And clearly that's been the clinical experience as well, that there are not major differences in terms of adverse events or infections across these different agents.
Got it. And certainly we will go back and address this table, whether it means deleting it or putting something else out there. In the final report, this table will be addressed. Okay, any other questions on the vote here? Okay, please go ahead and vote. Baricitinib plus conventional DMARD versus conventional DMARD alone. $14 yes, 0 no. All right. Okay. So now we shift to a different set of questions. Yes, okay, sorry. Oh, now I need to go back. Just to read to you. See if I can go back, actually. No, okay. Laura, can you come up and show me how to go backwards? That's the problem. I just want to tell them for what patient population we're working with. Oh, thanks. Okay, so for the following questions, we had to focus it in, and we decided to do it on patients, again, with moderately to severely active RA, who are TIM naive, that's where a lot of the evidence is, and who were failed by conventional CDMARDs, conventional DMARDs. So for those patients, think of them as kind of a stereotypical patient population, if you will. We're going to be asking about potential other benefits and contextual issues. So often these are things that lie outside the clinical trials. So here's the question. Does treating patients with upadacitinib plus a conventional CDMARD offer one or more of the following potential other benefits in comparison to adalimumab plus a conventional DMARD? So select all that apply. Let me just quickly run through them. So the first is, does this intervention, UPA versus adalimumab, so does UPA offer reduced complexity that will significantly improve patient outcomes? Does UPA significantly reduce caregiver or broader family burden, again, versus adalimumab? Does this intervention offer a novel mechanism of action or approach that will allow successful treatment of many patients for whom other available treatments have failed? Does this intervention have a significant impact on improving patients' ability to return to work or their overall productivity? And E is really an open question. Are there other important benefits or disadvantages that should have an important role in judgments of the value of this intervention? And again, we're doing a comparative here, so it's comparing UPA, these potential benefits, with adalimumab. So let me ask the patients, as you guys look and hear from patients and families, what stands out to you? And it may be E, it may be something that's not on this list. But certainly give us your thoughts on anything that's there, ways that you think decision makers should take into consideration some of these other potential benefits.
Sure. I'll kick us off. I think that
When comparing the method of administration between UPA versus adalimumab, you have an injection, a subcutaneous injection for the adalimumab versus an oral treatment that can hold lots of promise for patients. And so to that point, when it comes to administration of the drug, I think it does offer some improved patient outcome just based on those contextual considerations that you would consider, having a shot night versus not having a shot night, fear of needles, not having fear of needles, pain in administering the drug, all of those questions that link to adherence as a result. I also would put forward to the group a daily administration of an oral medication has some potential benefits when you're thinking in comparison to a product that you have to take once every 2 weeks or once every week. When it comes to having a biologic that works for the long time, what we hear from patients quite a bit is that once they're 5 days away from their next dose, they often have this tapering effect where they aren't as well controlled as they were right at the beginning of that, right after they had their drug. And so that consistency of delivery, I think that speaks to your ability to return and stay at work or stay at school or whatever that routine might be for you, as compared to an injectable product.
Can I just, before you go on, thank you, that was great. Clinical experts, while that's in our ears, do you also hear this idea of it wears off just before I take the next shot? Is that, because I bet you if the companies were up here, they'd say, no, that probably doesn't happen. So I'm curious about whether this is there any data on this, or is it just accumulated stories from the front, which doesn't mean that it's not data?
To me, it's more the latter. I've not seen any data speaking to that, but anecdotally, we hear that all the time with our weekly or our biweekly, with our monthly treatments, with our every 2-month treatments. It is a common story to hear, in the last few days or the last week that there's an increased number of symptoms. And, in general, I'll take the patients at face value with that and believe that it probably is a thing, but I'm not aware of any data.
Okay.
Yeah.
Fascinating. Andrew, what do you think?
I think that might be a reflection of the fact that medication is not adequately covering the entire interval, and that might be a sign that you're losing control or have lost control of the disease in that patient. So I would use that as a reason to be more cautious with the disease activity in the patient, because realistically speaking, the opposite is true as well. We see a honeymoon period whereby if someone feels ill around the time when they're due for their injection, don't use that injection during that week. Most often they get away with 1 week without doing it. Many get away with 2 weeks without doing it. It's rare to get 3 weeks, and almost nobody gets 4 weeks, but there is this opposite effect where they're controlled well enough that they can, in effect, extend out away from the typical timing for their injection. So to me, it's a sign that we might be losing control of disease if we're starting to have days without adequate control at the end of the dosing interval.
All right. And again, Peggy, you're coming. But one more question about this, because we heard with psoriasis, it tends to strike people at a younger age, that there was a fairly large number of adolescents and even college-age kids for whom this was a big deal because you could travel overseas, not have to worry about the storage, refrigeration, et cetera. I assume that's the same here, where oral medication just makes it easier to take that 2-week vacation. Or am I right? Do you hear that too?
Yeah, I'll ratify that statement. Yeah,
Okay.
Especially for people who are going abroad for 6 months' time, studying abroad, those types of considerations, that's a major quality of life benefit to having an oral medication. Yeah.
Okay. Now, are you ready for one disadvantage?
Sorry, can I just comment
please.
that, in that respect, sub-Qs are better than the infusions. And that the infusions are the worst in that scenario. And that if you're leaving, it's almost impossible to arrange. I say this from experience, to try to phone ahead and get an infusion set up elsewhere is virtually impossible.
Now, I wish we had data on this next question I'm going to ask, because it's a complete fiction in my head. But given the financial burdens on patients and families for these drugs, it would be easier to take your oral every other day or to split the pill as a way of nursing yourself through with a lower out-of-pocket burden than the injection, which you can't split in any real way. Do we hear any of that happening out there that is just something to keep in mind around value?
Don't be so sure people don't skip injections to save money.
For the same reason, yeah.
They'll be given a prescription that says do this once a week, and they'll do it every 10 days.
Okay.
Or 2 weeks becomes 3, or monthly becomes Q6 weeks. I would definitely agree with that.
Yeah.
Right, and stockpiling medicine absolutely occurs as well, right? So if for some reason you were ill, you didn't use an injection, or you were, heaven forbid, hospitalized and missed some doses, you have those doses at home to use for a rainy day, and they absolutely do get used in that way.
Okay.
Yeah.
Great. Peggy, please now come in. Oh, I'm sorry. Did you
Can I
want
add one more thing? I'm sorry, Peg.
Sure.
One additional contextual consideration is how you receive your medication in the mail. Oftentimes we hear with injectable products that when they are delivered that there's an issue with leaving the box outside in the sun for too long, or the truck wasn't refrigerated for the whole time, calling into question the efficacy of the product once it is delivered. With an oral medication, you don't have those same question marks at the end of when you receive that drug.
Okay. Please, Peggy, thank you for being patient. Any thoughts on this list of potential other benefits that you'd like to add?
One question that I would have, so I take an injectable, and when I feel a cold coming on, I skip a dose because you can't fight a cold if your immune system is suppressed. Duh. So my question would be, if you're taking something every single day, how do you know when to not take it?
All right. Any quick answer from the clinical experts?
I think it's a judgment call that potentially speaks to the advantage of the oral daily administration, having a faster on and off switch, so to speak. So typically in patients who we may see at higher risk of complications, infections, or otherwise, we have drugs that we administer anywhere between weekly to every 2 months or every 6 months, for that matter. Mm-hmm. So we would prefer the shorter half-life for those patients. So if you mess up and take it for a few extra days, you still get it out of your system fairly quickly when you stop taking it.
Okay. I'm sorry. Go ahead.
Sorry, lastly, with context, and I think this just speaks to patient heterogeneity, not to me speaking out of both sides of my mouth, but some actually prefer the infusions because it's every 8 weeks. Right. Come and get their infusion. They're done for 8 weeks. Don't have to worry about taking the pills. Don't have to worry about anything else from a medication perspective in between. It's all finished, wrapped up, and done for 2 months when they get that done. So that's also, represented in the population of opinion.
Yeah, and I'll endorse that as well, not only for the infusion, but also for the subcutaneous injection, that if you have a trouble taking a pill every day at the same time, that can be a benefit in the opposite direction.
Yeah, and if the family member or caregiver is giving it, it's easier once a week or once every other week than every day.
The other piece that I'll add while I'm on a soapbox is that the likelihood of an injection site reaction being a deterrent for a patient completely goes away with that oral administration. So that's another contextual piece.
Okay, great. Why don't we go ahead and vote on the potential other benefits? Oh, I'm sorry, Richard.
In answer to your first question.
Yes.
My personal PRO is when taking an injectable many years ago, I would find that it would taper off before the next injection was due. And with my doctor's approval, we would have the dose and take it twice as often. And then it became, as you said, Dr. Konkoff, the signal that it was time to switch. And the good news is that we now have I have psoriatic arthritis, same rules apply for RA. We now have a lot of choices which enable us to have the ability to switch, get a slightly different chemical mix as well. I have a question or comment. I think it's relevant to this question. I've been trying to figure out where it was relevant. And that's comorbidities. Mm-hmm. I was wondering to what extent those were considered in connection with the adverse effect analysis. Should we do A or B first and then move on to the next one, make it more complicated for you to answer? But B is, I'm very well aware that their patient population for for both RA, psoriatic arthritis, typically has 2 or 3, many autoimmune disorders typically have 2 or 3 additional autoimmune disorders, and other conditions, including high blood pressure. And fortunately for the patient population, the TNF inhibitors have a very positive impact on cardiovascular conditions. That was discovered by Nahal Mehta 8 or 10 years ago. I was curious as to whether any studies have been done on the JAK inhibitors relative cardiovascular, or is it too soon? Because that could have an impact on the algorithm that a physician will use in deciding which medication to use.
I'm not aware of any studies looking at cardiovascular outcomes in particular. You know, one concern both with the JAK and IL-6 inhibitors is this idea that they may raise lipid levels, which has been shown, although that's not been shown to increase adverse cardiac outcomes. But that's been a question that's been raised. I don't know that anybody has studied on the favorable side.
And we just, for completeness, should point out that although TNF inhibitors, by controlling disease activity, likely have favorable impacts on cardiovascular risk profiling, they are contraindicated in more advanced stages of heart failure as a result of trials that were meant to show benefit of control in heart failure inspired by TNF use, but which were stopped early for negative outcomes.
Okay, great. We need to try to keep going, so we're going to be really quick on this so that we can get through to the convention.
Pregnancy.
So there's only one, Tim, that's safe in pregnancy, and that's Cimzia certolizumab. It does not cross the placenta, according to studies that have been done, nor does it enter into breast milk. So the crib and cradle trials were a huge step forward for women of childbearing age, and in my estimation, because half of pregnancies are unplanned, women of childbearing age should have access to that drug preferentially to others, and yet that's not been universally the case and requires 20, 30, 40 minutes conversation with payers to make that clear. So to be clear here, only certolizumab is safe in that it does not cross placenta or into breast milk.
So in regards
and we will,
to JAK inhibitors, though.
Sorry?
No,
JAK inhibitors.
no data with JAKs in pregnancy we would avoid.
Right, and we will come back to sequencing and some of those issues related to step therapy, in particular in the roundtable. We okay? That's okay. Let's go ahead then and vote. Remember, all that apply, and I'm going to assume right now that there's no E because no one spoke up initially, so I hope that's the case. If there is, speak now. Otherwise, let's go ahead. Oh, Jeff, go ahead.
If we believe there's a benefit related to oral administration instead of injection, is that an E kind of thing?
That's an A kind of thing.
That's an A kind of thing because that's going to lead to better outcomes?
Mm-hmm. It's meant to imply that. It would be captured there by oral versus better outcomes on not just clinical but other elements. Liz?
Sorry, I know we're trying to...
Sure.
But this is relevant, I think, to what my question was in terms of what one is a bit semantics, but what is significant? Because clearly some people will prefer oral, some people will prefer injection. That's certainly my clinical practice in endocrine. Mm-hmm. Is that going to improve patient outcomes, maybe for a small number of patients, but is that significant? Right, it's not like sometimes these things are significant for everyone. So there's a subset of patients where this will be great and an improvement. But how do we?
If there's a subset of patients for whom you think it will be significant, and I hate to say it, but a significant subset for whom it's significant, I would go there because the purpose here is to be comprehensive. And so if you think that someone making a formulary decision or somebody else should know this before they kind of wrap it all up and make a judgment, then feel free to say yes to that element. We don't have a specific kind of quantitative line in the sand for that. Okay, so please do go ahead and vote. Now, since there's no fixed number of votes, I just need to look at you, and when you're done, kind of look up at me so that I know that you're done voting on this. All that apply. I'm still at least one person. Oh, you're all done. Oh, Annette's still voting. Okay, everybody done. Great. Oh, I had it on the, all right, let's try this again. There we go. Okay, 14 out of 14, A, 9 out of 14, B. You can see this, 3, C, and 6, D. Okay, given the time, we're going to not debrief on this and go straight into the contextual considerations. We're going to run a tiny bit over, but we can survive that. So here again, these are meant to capture kind of broader aspects of the condition or the treatment. So are any of the following contextual considerations important in assessing the long-term value for money at the current pricing of upadacitinib? Select all that apply. A, this intervention is intended for the care of individuals with a condition of particularly high severity in terms of its impact on length of life and or quality of life. It's intended for the care of individuals with a condition that represents a particularly high lifetime burden of illness. Compared to adalimumab, there is significant uncertainty about the long-term risk of serious side effects. Compared to adalimumab, there's significant uncertainty about the magnitude or durability of the long-term benefits. And E is the open to new ideas, additional contextual issues that should have an important role in judgments of the value. All right. I think we've been talking, go ahead, Alex.
Just this question is directed towards Jeff. In Table 21, I'm interested why C was not selected. Oh, no, not C. Let's see, is it C? The uncertainty of the long-term side effects is less well known for upadacitinib. I'm not.
UPA. You can say UPA.
Yes, UPA compared to adalimumab.
No, I think that's a reasonable.
Is that reasonable?
Yes.
Okay.
When we ask that question, it's obviously meant to capture some specific some specific scenarios. Some people, it will matter whether it's the same mechanism of action as a drug previously tested, and therefore, even though it's brand new, you have more certainty than you would if it was brand new as a mechanism of action. You've heard that it's not the exact same receptor sites within the IL pathway as some others, so all of that, I'm sure, will factor into your thinking. All right. With all due respect, I think we're going to move forward with the voting here so we can get to the value question, which in a sense integrates this. But please feel free to raise your hand if you have a strong question or statement to make otherwise. Okay, please go ahead and vote, and then look up at me when you're done. Kim, are you still voting? You're done, okay? I think we're all done then, everybody? Okay. Here we've got 12 for A, 12 for B, 4 for C, 5 for D. Okay, this will all be captured in the report. As we move to the final question, right, we're back again, staying, I should say, with naive to Tim's. And now here's the value question that we have. The preamble is kind of long, but again, given the available evidence on the comparative effectiveness, which we've talked about and voted on, the incremental cost effectiveness using the net price estimate of 26% off the wholesale acquisition cost. Just to remind you that this was a placeholder price based on average rebates in that class. So given that price estimate, and considering the other benefits, disadvantages, and contextual considerations that we've just spoken of, what do you feel is the long-term value for money of treatment with upadacitinib plus a conventional DMARD versus adalimumab plus a conventional DMARD? Is it A, low long-term value for money at this estimated pricing? Is it B, intermediate long-term value for money? Or is it C, high long-term value for money at estimated pricing? And Rick, the one piece of information that you might want to hear again was the incremental cost effectiveness ratio at this estimated price for UPA versus ADA.
Yeah, so that's shown in Table 16 in your evidence summaries. For the base case, it was $92,000 per quality gain. And if you look at Table 17, you can see the results of the probabilistic sensitivity analysis. which showed 54% of simulations under $100,000 per quality and 80% under $150,000 per quality.
Got it? $92,000 per quality. What was it per life year gained? Probably was on a year model. We didn't
do
that
life
one.
year because it's a year.
So think of 92,000 per quality. All of the other issues that we've just spoken about. Let that just sort a little bit into your head. Questions or thoughts on how you feel you are integrating this information before you vote? Low, intermediate, or high? Anybody want to jump out in front and say, I'm going to vote X because this, that, and the other? You know I'm going to pick somebody. Jeff?
So I'm sort of looking for advice. I'm kind of between, I have a colleague who thinks it might be medium, and I have another colleague who might think it might be low, and I'm trying to advise them about how to vote. So I was wondering if you could give me any help on that. The person who thinks it's low is thinking that if you look at not only the other contextual and the other benefits, if you also look at the cost effectiveness, there seems to be not a huge amount of extra qualities that are happening, although it's only been for a year. And the person who thinks it's moderate value thinks that, yeah, that might be meager. However, there's a lot of other things going on that we might interpret as value. So I need some help from the committee. I don't know what to tell them.
In the words of a great clinical expert, yes. That's why we pay you the big bucks and feed you the cheap donuts. So you get to decide. Liz.
So I'm
Lisa,
not the economist, but so I can't be your colleague. But my concern is a little bit about the clinical advantage that almost all of us voted and said was there, is clouded by that C-reactive protein issue and the subset of patients who are in on that. And so when you look at applying this to more across the board, I think that my confidence that this is going to be advantageous across the population goes way down. Okay. And so I wish we had an I for not enough evidence or inconclusive.
Thank you. That's very helpful to hear people's thinking. I've got Neil, and then I think Say wants to speak as well, perhaps.
I wonder just for context in other disease states and other interventions, in previous discussions I've seen cost per quality in the range of 100,000 to 150,000, and I think even historically that has bumped up over time, and that many things that are kind of in the mainstream are in that range, but I want to make sure I was understanding that as a cost per quality as a comparator.
So there's both we and others contribute to this conversation around the world, and there are different approaches to thinking about ranges and thresholds. We have proposed in our current update to stick with 100 to 150,000 as a general range. Now, anytime you start to get to precise 92 versus 93 versus 94, it's usually not worth making those kinds of distinctions. But that's just a piece of the decision. So others include uncertainty. Some people believe if there is a certain amount of uncertainty about the generalizability or the long-term effects or risks, then you would want a lower threshold in order to feel confident that you're getting reasonable value. Other people don't think that way. There are some people who feel violently attached to a lower threshold to begin with. They think that 100 to 150 is paving our way to bankruptcy, and others think that it's woefully low given Americans' known predilection to spending on healthcare. So you can hear those in different ways. We've tried to peg a threshold range at which there is some evidence that things priced beyond that lead to, in a sense, increases in healthcare premiums that lead to increasing dropping of healthcare and delaying of care that actually have negative health benefits. And so we're actually trying to figure out what's the range at which we maximize the health in the overall system, recognizing that we know that we're gaining benefit from the patient in front of us who receives one of these treatments. But we just can't see the consumers, the other patients who are grappling with the costs and making decisions that may negatively affect their health. So that was another kind of non-answer. I tried to give you multiple different perspectives on it, but it's a good question. Say.
So I will also provide a non-answer, arguing both sides before telling you what I am going to say. So I am most concerned about the comment from our clinical experts about the proportion of RA patients who would be excluded from the data that we are. So we are, and this is not a random subset of RA patients. And if I am making the assumption, not being a rheumatologist, that these studies are like all the other studies that have been done, where the people being studied are generally younger, healthier, of a monodisease cohort, and we are going to be making a judgment now and extrapolating that to a much larger, older, sicker, more comorbid population. And our experience with other conditions have been that when we move from a RCT population to a real-world population, the net benefits go down. And we are already looking at fairly marginal benefits. And then when I think about, okay, we are talking about the benefits, the numbers that we're looking at are from this small population of the ones who are most likely to benefit. And we are probably letting loose this intervention on a much larger population where the harms are likely going to be greater and the benefits are likely going to be smaller. Now I'll argue the other. I do think that it is it's important for us to think about the heterogeneity of the population. And although I recognize that this is one of many classes, many subtypes of immune modulators, the JAK inhibitors feel a little bit different. And when we talk about RA patient populations, I think we're talking about smudging together many genotypic subtypes. And there are going to be some patients who probably are going to be much more responsive to this medication than anything that is currently out there. And so I do think if we look at average responsiveness, it may be quite small, but for individual patients, there may be ones for whom this is actually an incredibly efficacious and cost-effective option. So I think for me, it feels like that is a very strong consideration, making me, otherwise I would vote hell no, but that makes me that sways me to give it a more favorable rating.
Okay, great. Now we do have to move forward, so I'm going to try to let you guys have a couple more comments before we vote, but try to make it as brief as possible because we're already eating into our break time. Bob.
I want to make mine shorter than usual. I'm going to vote intermediate because I don't think it's decidedly low. I think we're all in agreement it's not decidedly high. To me, intermediate is an important vote here because that's our one chance to express our actual feeling about this and to help encourage the industry to drive that 26% discount higher. I do want the prices of these things lower. They're not that advantageous price-wise. They could be more advantageous, and I would like to do that in the guise of this doing our part that we do to encourage more innovation in this area. So.
Thank you. Annette?
Yeah, so I think the one thing that makes me uncomfortable is that there are no head-to-head. One, it's a small study, right? The head-to-head trial. It's a single study, and there are no head-to-head trials between the JAK inhibitors. And so it does make me a little uncomfortable. If you look at the placebo group, it's healthier in this trial than it was in the TOCA trial. And probably a lot healthier than the ones in the TNF-alpha inhibitor trials. And so it does make me uncomfortable with the evidence that we were really getting into something that's intermediate and high, so I am actually going to vote low on this.
Just to note, the question is specifically framed of UPA versus adalimumab.
Yep.
So it's just want to make sure that you saw that part.
There's a whole class of drugs,
right.
and only one shows superiority to Humira. Okay. And that makes me uncomfortable, and it's only a single small study.
Okay.
So.
Rita, you get 1 second. Okay, then we're done. No.
So I agree with a lot of my, Annette, and say for sure on the concerns about the trial, but I will also say, I'm very concerned when we start talking about discounts and coupons and out of pocket, because that's just playing around. It doesn't do anything to affect the drug pricing. It allows you to keep drug pricing really high. It's still a big burden for patients, and I think, with everything we saw, I feel this is a low value.
All right. I think you've heard a good gamut of both votes and rationales behind them. Apologies. You may have. I'm sure you want to vote. Part of you wants to vote. We usually, again, as you know, our feeling is that putting, especially patient representatives, in the place of voting, because this is your population, and it's completely natural to feel like the value clinically is what really matters. And I think you've done a great job of expressing that to people in the room. So let's take a vote integrating all these considerations. There should be 14 votes, and I will show you the results. 8, low value. 6, intermediate value. And 0, high long-term value. Thank you for the great conversation before the votes, so that I think it was very healthy for people to be able to hear that. We are going to note this, and we are going to move. We are going to be able to have to take still a 15-minute break, but that means we are not going to go 20. So right on the dot at 3:30. Thank you. Right on the dot. In some part of the world, it's 3:30. At 2:30, we will launch right into our policy roundtable. We are going to tackle rebates, biosimilars, what the heck's going on with step therapy, what can we do to make the world a better place. 2:30. Thank you.